Good afternoon. Good afternoon to all. God provides us with herbs for the service of man. With this quote, I, Sarita Singh, Assistant Professor, Institute of Pharmacy, Shri Ram Sarup Memorial University, welcome to all our dignitaries, faculty members, and research scholars on day 6 of the FDP on Advanced Research Methodology in Pharmaceutical Sciences. Today, the topic will have focused on recent advances of phytopharmaceutical research. For this, I would like to welcome our special guest and speaker of the session, Dr. Alok Mukherjee sir, Principal at United Institute of Pharmacy, Allahabad. But before the start of this session, I would like to brief about Dr. Alok Mukherjee sir. Sir has a work experience of over 25 years in the field of pharmaceutical sciences. Sir has awarded his PhD in pharmacy in the year 2009 on the title Phytopharmacological Activity of Some Herbal Drugs for its hepatoprotective activity. Sir has been invited as a speaker and chairperson in various international conferences and national seminars, and invited as a resource person in various AICT and AKTU sponsored programs. Sir has research publications in various national and international reputed journals. Sir has over 23 international research publications, 41 national research publications, 16 Scopus Index publications. Sir has published a book entitled Traditional Medicine for Herbal Plants, sorry, Traditional Medicine Plants for Hepatoprotection. Sir has wrote various book chapters in the topic such as Global Approach for Drug Discovery and Development from Indian Traditional Medicine, HPTLC fingerprinting analysis of phytoconstituents from indigenous medicine plants. Sir has been a member of various board of studies such as Assam University, VB Singh Purvanchal University, Jaunpur, Dr. APJ Abdul Kalam Technical University, Lucknow, CSDM University, Kanpur, SHIATS AAID University, Allahabad, MJP Rohilkhand University, Bareilly, UP. Dr. Alok sir has been selected as the various paper setter and external examiner, and even selected as a PhD thesis evaluator in various universities like BHU, Banaras Hindu University, Varanasi, Dr. APJ Abdul Kalam Technical University, Lucknow, BBD University, Lucknow, Sam Higginbottom University, Schwartz, Allahabad, Chitkara University, Punjab, CSGM University, Kanpur, MJP Rohilkhand University, Bareilly, VB Singh Purvanchal University, Jaunpur. Sir has been a supervisor of around 32 MPharm students, and 11 PhDs in pharmaceutical sciences has been awarded to students under his guidance. Sir has a life membership of Indian Pharmacy Graduate Association IPGA. Sir has been a life member of UP State Coordinators of Indian Society of Pharmacognosy, Association of Pharmaceutical Teachers of India, International Academy of Physical Sciences IAPS Allahabad. Sir has been a vice president of Uttar Pradesh State Branch of Pharmaceutical Education Society, originated by teachers and students, PESOTS. With this note, I welcome Dr. Alok Mukherjee sir to start today's session. Sir?
Good afternoon, everybody. Can you hear me?
Yes, sir.
Am I audible? Okay. Allow me to share a screen. Can you see my slides?
Yes, sir, it is visible.
Okay. So, uh, thank you for giving me the opportunity. I'm really thankful to all the staff members, those who are part of this Pharmaceutical Institute, and especially Dr. Manju, who is the principal of the college. And I'm definitely in depth. Thank you for the management of this Ramesh college, and that's a very nice opportunity for me to be a part of this FDP program. I always think that these are the things to be developed, some of the teaching methodology, some of the new researches, some of the new technology can be learned through a teacher. And this teacher, if learned something, then definitely it will be transferred to the our new budding institute, and also the students of the pharmacy when they go through a teaching offline or online meeting. मेरे ख्याल से मैं जब ये डिस्कस कर रहा हूँ तो सारे टीचर्स ही होंगे इस एफडीपी में, सरिता जी।
Yes, sir. Sir, research scholars are also there.
रिसर्च वर्क पर।
Faculties and research scholars. Yes, sir.
अच्छा, definitely, definitely. So welcome to all research scholars also, those who are starting their career in the field of this PhD or other part. I'm definitely trying to give you some short view of the new research development, where we can have a certain identification how to go for a research, especially in case of pharmaceutical research, where I have a specialization of pharmacognosy. So that's why I had little bit glimpses of this phyto-pharmaceutical research, where the herbal drugs are being a major part of my topic. Okay. In between, anybody, if you want to ask anything, definitely you can raise your hand and discuss with me. uh, Ask to this master of ceremony to open your mic so we can discuss the thing. It could be an interactive session. So anything you want to discuss, anything you want to ask, let me discuss, and so we can discuss the thing very nicely. So today, my topic is recent advancement of phytopharmaceutical research. That means whatever research we are going through in the field of pharmacy, they must require some of the betterment of science, betterment of the pharmacy, as well as the betterment of the future of the pharmacy student. So my topic should be much more related to the what are the new things are being developed in the field of pharmacognosy research or the herbal drugs especially. But before that, we also like to share some of the glimpses. What are the different requirements, what we feel in case of teaching, definitely in case of industry requirement, what the industry has required, where we are lacking, and how we grow this herbal industry research especially. So my topic should be multidisciplinary. It can be directly to the part of manufacturing. It can be directly related to the teaching. It can be related to the how you study the phytopharmaceutical research especially. Okay. Traditional part that I look here, this is a very important thing that I want to discuss with you. In traditional system of medicine, especially Ayurveda, Unani, Siddha, and we call in India, which we call Ayush, the plant, minerals, and animal products are being used for the main active ingredient, and they are basically used for the curing a different type of ailments. Medicines are very widely throughout the global useful, and people are rely on that. And this relying on the Ayurvedic or Unani system, or the you can say the alternative system of medicine, definitely makes our responsibility toward the society how we can cure the different type of ailments to the patient or to the public. So the practice of traditional medicine vary greatly from country to country. It may be in India, the different practice are there. In case of China, Those are a developing country or developed country. In case of Europe, there are different type of practice of traditional medicines are there. They can use directly as a medicine. They can call as a herbal medicine. They can call as a alternative medicine, or sometimes they can call as a food and medicines for uh, healthiness, wellness, you can say. And region to region, it can also vary. So the requirement of the herbal medicine may vary country to country and region to region, and depending on the how they are feeling with this traditional medicine. Herbal drugs may vary in the composition and properties, unlike conventional pharmaceutical products, which are usually prepared from synthetic, chemical, pure material by means of reproducible manufacturing technique and filtering. Because whenever we discuss the comparison between the modern medicine and the herbal medicine, the compositions of modern medicines are completely depending on the synthetic preparation because they are the purely chemical compound. So they can be easily reproducible, and each batch to batch variation is very little. But in case of the herbal drug, the compositions and properties are completely vary because every time it is being used on the basis of what are the herbal products are. And these herbal products
कि हम जो है
can
पेपर डालेंगे ऑनलाइन डाल देंगे
vary
देखेंगे
region to region, nation to nation, especially on the basis of geographical distributions also. The same plant which has grown in UP may have a different biological and chemical constituents compared to the different other states in India especially or in case of region to region also. So the composition and properties in case of the alternative medicine are not very defined compared to the modern medicine. But there is a global resurgence in the use of these medicines along with the growing scientific interest in them as a source of new drugs. So we have to have, being a pharmacognosist, being a pharmacist, we have to think about that how to rely on the patient, how to make our responsible scientific research to benefit of the society. The use of well-established and widely acknowledged as a safe and effective and may be accepted by the nation authorities also. And that is why most of the nations have started the alternative medicine as an important medicine in their field of pharmacy. Herbal preparations are produced by extraction, we all know, fractionation, purification, concentration, or other physical and biological process by heating herbal medicine in alcoholic beverages or by any other, uh, you can say, solvent like honey and materials also. So these preparations are really when we take the different part of the herb or the plant, then we can go for the extraction and fractionation and then purification and concentration. It may be by different techniques of the extraction methodology can be used. As humans have mastered the technology of drug synthesis, plants remains a good source of drug discovery. So our main aim in the research methodology that how we can take that particular compound which are being useful for the treatment of any ailment can easily be collected from the plant and can be isolated from the plant and become a good source of drug discovery. At present, more than 40,000 plant species are used for the medical purpose around the world, or it may be a much more than it is not known scientifically. In conventional Western medicine, 50 to 60% of pharmaceutical commodities contain natural products or be synthesized from them. That means if you can just convert the total world, whole global type criteria and find out that 50% or 60% of the pharmaceutical commodities are being having either single natural product or we may be a multi natural products are being there or they can
Sir,
be synthesized from them.
sir,
Is there any question?
sir,
Yeah.
sir, slide is not moving, sir.
अभी किस स्लाइड पे हैं हम लोग?
Sir, it is second slide.
अभी ये मूव हो रही है?
Uh,
अब
no, sir. No, sir.
अच्छा, एक मिनट, मुझे लगा मूव हो रही है। अच्छा, अभी बताइए, पहली स्लाइड दिख रही है?
Ji, sir.
अब
Yes, sir. It's moving.
इट्स मूविंग ना? ओके
Yes, sir. Now it's moving.
ओके, so I'm just revising the things. We can start from the traditional system of medicine in India, which are Ayush, we can say, which include Ayurveda, Unani, Siddha, and Homeopathic system. And these, all the alternative medicines have a plant, mineral, or animal as a origin, as a natural source, which can be useful for the different ailments. And this is the part that every traditional medicine, either in India or in China or in throughout the Europe or different countries, they are using this type of traditional medicine for the betterment of the life of the people, and it can be very useful for the society, social mm, development of the society. The practice of the traditional medicine vary greatly from country to country. As I earlier said, that in each country, either it is in Europe or in case of China or India or in different parts of the Asia, the practice of traditional medicine vary. And that variation in that way because the plant origin or the herbal origin drugs are also vary. The initial ingredients of the plant which develop at India may be vary in case of China, may be vary in case of Europe. So the practice of the traditional medicines, starting from the India to different other countries, vary country to country or also region to region. In India also, if you are in Rajasthan, if you are in Gujarat, or if you are in UP, the country, the different geographical region, the plant can vary, the different plant species can vary, and that is why the practice of the traditional medicine is also that vary. Slide is moving now.
Yes, sir.
Okay. So, as I talked earlier, that herbal drugs may vary in the composition and property, unlike the conventional pharmaceutical product, because the modern medicine having a chemical synthetic product or chemical compound, and they have a reproducibility during the manufacturing technique. That means in every time when the modern medicines are being manufactured, the batch to batch, they have a simple same type of QA and QC analysis. But in case of herbal drugs, which is having a composition compound, and the compositions or the properties can vary due to the raw materials from different source, they may have a different type of product every batch to batch. But that will never be harmful for the patient, those who are using it. So herbal drugs compared to the modern medicine may have a vary in composition and property also, which is not possible in case of herbal drugs. Although there is a global resurgence in the use of these medicines along with the growing scientific interest in them as a source of new drugs. So throughout the world, in different regions, the people are now rely on this herbal medicine due to have a lesser side effect, and having a benefit of the different disease where they can useful or betterment for the society. As I said, that herbal preparation can be used or produced by extraction, fractionation, purification, and concentration. And this is a basic requirement for every researcher to know the quality of the herb, the part of the herbal plant, where, how to go to extraction, what are the procedures of purification and concentrations are required, because they need a different type of knowledge of solvent as well as the compound present in that plant. So herbal preparations are produced by different techniques, and most of the techniques are maybe extraction or the fractionation, and there are different type of purification methods are also there. At present, more than 40,000 plant species are used for medicinal purpose around the world. In conventional Western medicine, 50 to 60% of the pharmaceutical commodities contain natural products or they are synthesized from them. That means we can say throughout the world, half of the pharmaceutical commodities are made by natural products or maybe synthesized from the natural products. That means most of the herbal drugs are being a part of the whole medicine throughout the world. More than 25% of all prescription drugs contain one or more ingredients derived from the plant. We know, like paclitaxel, vincristine, vinblastine, artemisinin, they are very popular drugs, podophylotoxins, camptothecin. These are the very popular drugs and they all are derived from the plant. That means whatever the different companies, whatever the modern medicine we are using nowadays, they may have directly or indirectly completed from the plant or isolated from the plant compound, and then they may be semi-synthesized or synthesized from the plant. Voice is clear.
Yes, sir.
Slide is moving now.
Yes, sir. It's moving.
Okay, thank you. So this is just a herbal formulation part because why the people are rely on herbal formulation because they are safe. They are eco-friendly. They are little cheaper than the other modern medicines. Less adverse effects are there, and they are having a permanent cure. So, herbal formulation having much more important in our life day by day, and thereby they are being much more useful towards the patient element. But our major part is to be discussed here today. So, how to make this herbal drug in a better way that it can be beautifully useful for the different type of ailment, which cannot be cured by the modern medicine, or they can be parallel to that modern medicine also. For this, we need to have a drug discovery. And drug discovery strategies based on natural products and traditional medicine are re-emerging in the anti-science choice of new drug candidates to facilitate discovery process and for also for the development of rational synergistic herbal formulation. That means our aim is to make a herbal formulation which is much more useful and it can be a lesser in quantity but having a better quality. Because if you take a simple herbal formulation, the quality of the product may be lesser and the quantity of the dose may be more. But if you go and you convert into just this chemical compound, isolated compound, a very important compound, then they can be very widely useful as compared to that earlier slide I said the paclitaxel useful for the cancer, vinblastine useful for the different type of cancer, and these are the drugs which are being useful for the modern medicine and modern society also. So nowadays, pharmaceutical companies currently emphasizing their exploitation in the new drug discovery by screening and characterizing instead favor synthetic compound as more tractable alternative. So you can, if you go through a different pharmaceutical company, they have their own sister concern also, where they have discovered a new modern medicine from the herbal origin or the natural origin, and they are using their economy from these natural medicines also. Although they have their own product of modern medicines also. So the modern medicine and natural medicines are comparatively are quite good in a different pharmaceutical company. You can start with the Cipla, you can start with the Cadila, you can start with the Dr. Reddy's. They have all their, although have their modern medicine manufacturing unit and R&D also. Simultaneously, they have their own natural medicine R&D and natural medicine manufacturing process of unit also. If you compare with the total CAGR growth rate, consumer spending on increased to $7.5 billion, and this rising demand will further boost into Ayush and alternative medicine industry growth in India, expected to 8.6% in the year 2020 and 2026 within this 5 years. And this is a market summary. So you can say that the modern medicine is there, but alternative medicine are day by day growing their advantage, and they are getting a very important part in the life of the patient, or the modern local patient. Global herbal medicine market value 84.5 84.5 billion in 2019 and anticipated to grow 20%, 20.5% during the forecast period, I think at 2022 or 2023. According to the this global newswire, the market expected to reach valuation of 411.2 billion by the year 2026, and in the 2030 it may be a much more 18.9% CAGR. That means you can say if you go through in the market scenario, you can find out that day by day alternative medicine getting a value, and we feel people as a researcher, as a pharmacist, you can feel pride that herbal medicines are always being ahead in the coming era. Now I'm coming to the part of how the plant drug can be isolated. This is very simple. You can say the traditional medicine can be identified or selected. Different plants can be identified. It can be selected on the basis of their knowledge of text, old text, or you can have a traditional knowledge from the different Vaidya or different Ayurvedic practitioners. Then, after selection and identification by different methods, you can start with the morphological or anatomical identification can be useful. And by this, we can primarily screening to support the Ayurvedic claims, whether the drug has been useful or not. So whatever the old text or the old people has known about the traditional knowledge or the drugs, they justify it by their text or by knowledge. But we have to go for the primary screening, and this primary screening need to be a phytochemical profiling or high throughput screening. That means modern technologies are there, or you can say a simple phytochemical testing are there. By phytochemical testing, we can go for isolated bioactive compound or screen the theoretical potency. So this all a part of our pharmacognostic syllabus, and we have gone through this part in the field of master's degree or the research scholarship. This we all have done or we are doing in the process of identifying the Ayurvedic claim, whether the drug has been useful, whether the drug having this type of compound or not. The Indian system of medicine providing the drug with the cultivation collection to the its screening of the drug. Then after that, we have to go for the standardization control, pharmaceutical and pharmacokinetics, pharmacovigilance, and undergone a regulatory affairs and guidance. And finally, we can find out the drug of standard quality, which is useful for the clinical trial. So this is the compound comprehensive study of any herbal drug which is being not scientifically screened. Only the old text or the Ayurvedic text can say that this drug is being useful for the purpose of certain ailment. Then we have to follow this chart and go for different type of techniques to identify that particular compound, isolate the compound, then go through a proper affairs and guidance, and then it can be useful for the different phase of trials to betterment of the any type of disease. Okay, but nowadays this is the old system, but nowadays the process has been modified. Now the process of modifications required this process, which case that pre-drug stage and QC drug stage. Pre-drug stage and pre-study stage is definitely a different, where we can justify how the drug has been useful for the plant research. Is there any question regarding this part? I'm just describing this thing. The pre-drug stage is mean what we have to go for any herb or any herbal preparations, which then the traditional medicine system or the folk medicines or the practical experience or different type of food stuff has been called as the drug has been useful. Then we go for the extract isolation, and this extraction will guide you whether the drug has been useful for the any treatment or not. We shall go for the phytochemical isolation screening, and this phytochemical screening will provide you whether the drug has different type of chemical compound, like it may be having alkaloid or the glycoside. So different type of glycosidal test, different type of alkaloidal test, tannin proteins are there, and then we can identify the drug can be useful. So in case of that, there are certain other processes are there, like plant cell culture. This plant cell culture can also be useful for the purification, and then we can go for a isolation of a certain compound, and these isolation of certain compound can again give you the preclinical study drug and the clinical trial, and then go to the market. Okay, so herbal to synthetic or herb to herbal. These are the two different other process by which we can make this hybrid compound, and hybrid compound again go for the full drug stage. So there are three stages: pre-drug stage, quiescent stage, and full drug stage. And this whole process of any researcher need to know how to work on this phytopharmaceutical for any isolation compound to make it better and better useful for the society. I'm just elaborating this part. Pre-drug stage encompasses the information-driven selection of the house or the plant in the first stage of drug discovery. That means it gives you the first point that the herbs and plants must have certain compounds and how to go through identification of this. Like in case of Rolfe alkaloids, Sorelen, Holaria, Holarhena alkaloids, Gugolosterone, Becoside, Picroside. These are the very common compounds isolated from the different plant. It may be from Rauwolfia serpentina, it may be from Curcuma longa, it can be a Bidania somnifera, it can be artemisinin. Many other steroidal lactone and glycoside have provided a fruitful outcome in the area of drug discovery, drawing on Ayurvedic experimental data. So this to be required in case of pre-drug stage. With the advancement of analytical technology and biological science, many biotic compounds or the chemical entities have been identified through these plants or the different type of food stuff by the pharmacological and phytochemical studies. Like Taxol is an important anti-cancer drug, is isolated from the specific uses. Lutein can be isolated from the different marigold plant. Then lycopene from the tomato can be isolated. So this pre-stage will give you the identification that what are the different compounds are there and how it can be extracted from the different part of the plant. Then comes process drug stage. This process drug stage is nothing but a part of drug discovery from herbal medicine, include the preparation of extract and phytochemical group from herbs, including the discovery of lead compound by using modern internal research tools. Here we can use different type of techniques like HPLC, HPPLC, MAR, NMR, and justify that different chemical nature of the drugs which can be useful for the different type of treatment. Herbs contain hundreds of active ingredients and that may be potentially useful for the development of therapeutic agents. So the identification and isolation of phytochemical groups and single compound chemical interactions from herbs and the plant material are therefore crucial for the discovery. So every time we have to think about that, the single compound may not be very useful, but the plant having extract, having a plant, having a much more, more than the different compound, they may have a better effect on the particular ailment. So it is to be justified by the pharmacological activity of the drug that the single compound is more useful or the multi. So these are the this is very important techniques nowadays in the modern technology where we can use into herbal technology. The drugs can be either are extracted by the technique. Now, with the growing market herbal medicines, many countries have provided some regulation guidelines to ensure the safe use of medicine by the patient or the consumer. So in WHO 2018, about 90% of the member state and country have had national regulation on herbal medicine. So this is also very important before going for any research in the herbal market that we have to think out what are the different regulations are required. Earlier we have discussed about the extraction isolation in the modern technique. Now you need to know the what are the different regulations are they required for the any type of research we are doing in case of herbal medicine. Like reported to the use of national and other pharmacopoeia, especially those in case of India, Britain, Europe, and United States. All pharmacopoeias are there, and their monograph included the herbal medicine. So we have to find out the first different countries pharmacopoeia, and their report has to be find out. Provide guidance for good manufacturing practice and mechanism to ensure compliances with the manufacturing requirement. Enforce special regulatory requirement for herbal medicine or the similar requirement for those for conventional pharmaceuticals. So these three important parts are to be required for the regulation when we go for the research in case of herbal medicine. These regulations and policies are made and put into the practice to create a standardized quality of herbal medicine and ensure their safety and efficacy. So whenever we go for the research, if we go for the publications also, so we should know the what are the minimum requirement for the regulations of different other countries also. Whenever we go for any manufacturing or any formulation product in India, that should be follow the regulations of other country like in Europe, in case of Britain or United States. So these are the mandatory requirements for any research or the manufacturing part. Now, once we know the regulations, we have to find out whether our product is correct or not. That means correct identification and quality assurance of the starting material is therefore an essential prerequisite to ensure the reproducibility quality of these medicines, which contribute to the safety and efficacy. So whenever we think about the safety and efficacy, the reproducibility, as I earlier said, that herbal medicine having a multi-compound process are there. So we should be very careful about the drug discovery method that it should be a quality product. The quantity and quality of the safety efficacy data on the traditional medicine are far from sufficient to meet the criteria needed to support it worldwide. And that is the major lacking in this. Because every time same compound, same reproducibility is very less in case of herbal medicine. Why it is required? Because lack of research data, not only to the healthcare policies, but also the lack of adequate and effective research methodology for the evaluating traditional medicines are there. And for that, we researchers have to follow all the methodology and have to develop new techniques for evaluations of these herbal or traditional medicine. So that is our responsibility being a pharmacist, we have to be very careful about the different research methodology. For that, we need a proper literature review. Whenever we go for any research, the research review, literature review should be very important, and that is required by the every researcher, those who are in teaching or in a, you can say, the part of industry. Literature search should include reference books, review articles, synthetic surveillance of primary sources, and database searches. So this database is very important nowadays for any type of research publication, any type of literature review is there. Now, this is an approach for therapeutical agent. It is having a four part. First is drug discovery for the herbal medicines. The typical drug development process of herbal medicines include at least four different aspects. First is isolation or artificial synthesis of bioactive compound in herbal medicine. That means drug of a small molecule from herbal medicine can be synthesized or can be isolated. So hybrid molecule like synthesized by semi-synthetically or when isolated from the herb can be developed. But again, the safety and efficacy is required. The first is isolation or artificial synthesis. Second is evaluation of safety and efficacy using system pharmacological method. Here you can say the system pharmacology is followed, where biological database and disease database are to be required. These data sets are very important for the system pharmacology, and we have to process the safety and efficacy also. So first is isolation, second is system pharmacology. Third, evaluation of safety and efficacy by means of conventional pharmacological methods. So whatever the conventional pharmacology methods are there, that evaluation is will be very important by the basis of pharmacodynamics and by the basis of pharmacokinetics. So on the basis of the toxicology, pharmacodynamic and kinetics, these conventional pharmacological methods gives you the new approved therapeutical agent. And this is called regulatory approval of the therapeutical agent used in the market and post-market monitoring. So starting from the simple herbal drug isolation, we can reach to the new approved therapeutic agent by process by 1, 2, 3, 4 stages. And these are the four stages nowadays for the system pharmacology and drug discovery method of herbal medicine. And every researcher thinks about these four steps to how to go for a better bioactive compound. For any raw material to authenticate, we have to go for a very simple phyto-screening techniques: taxonomical method, microscopical method, and microscopical method. Morphological method and microscopic method and taxonomical method are very important for authenticize the herbal drug. So whenever you go for any research, you have to very careful about the identification of the drug and its taxonomical authentication. So while collecting a drug, you should be known that your species and genus is quite good, and it can be approved through a certain biological survey or any botanical survey of India. They are district wise or state wise. They have a different offices are there where we can have authenticate the our drug content. They can identify the drug by the taxonomical method, by morphological method, its texture, its content, its leaves, roots, and then also they can identify by its microscopic method. We already gone through all these things in case of our syllabus of B form or M form, where we can go for a authentication. Why it is required? By because the adulterations are much more used in case of today's herbal system. Like one of the example I have taken, this is the original drug called black pepper, but it is adulterated by a papaya seed very commonly. Dried papaya seed in black color can be adulterated, can be used as an adulteration in case of black pepper. It is a costly material. Similarly, in case of saffron, Crocus sativa, kesar, this original drug can be replaced by a safflower Catharanthus tinctorius, and this is a very widely used by the people those who are in case of adulteration. So saffron is adulterated with the colored stigma and safflower, and colored paraffin wax is used in case of beeswax. These are very good examples of adulteration. Inferior quality material like belladonna leaves are substituted by Ailanthus leaves, papaya seed adulterated with piper lime. I have given us 3-4 examples of this adulteration, and they are very useful, very harmful for the drugs or the publication in case of research. Whenever we go for any publication, people can ask whether we have gone for the authentication of the drug or not. And these authentication is much more required nowadays, whether you have to collect the drugs from market, whether you have to collect the drug from any natural sources or any folklore medicine source. So while knowing this adulteration, you should know the process of evaluation. So evaluation of herbal medicine is more important, by which value addition to the traditional medicine can be possible. Okay, and this knowledge of evaluation and adulteration is being shared with the doctors and the patients also, because whenever the doctors and patients are using this drug or prescribing this drug, pharmacist is the much more aware of all these things, because how to collect the drug, how to cultivate the drug, this all process is a part of the adulteration and evaluation. So better the knowledge, better you can give the value of the medicine. And for this, different type of pharmacopoeia standards are there. Food and Drug Administration, Ayush, and other regulatory bodies are there. Time to time, they are awareing you and giving the text to identify the different type of adulteration and how to evaluate them. It is also very important. Getting my sound? मेरी आवाज आ रही है?
Yes, sir.
Okay. Sometime कोई बोलता नहीं तो मुझे लग रहा है जैसे कि मैं अकेला बोल रहा हूँ. ये मोनोटोनस टाइप है ना. चलिए. So the quality control and quality assurance in case of this herbal medicines are very important, because we need to investigate the bioactive phytoconstituents and their analysis. जैसा हम लोग मॉडर्न मेडिसिन में देखते हैं, क्वालिटी कंट्रोल और क्वालिटी एश्योरेंस बहुत वाइडली यूज होता है, बट इन हर्बल मेडिसिन these process are being not being very important and very legally important required. So in every drug or every company have started their phytopharmaceutical formulation without using this quality control and quality assurance, and they come into the market. But being a pharmacist, we have to take care of this quality as an assurance and the quality as to control also. For that, different type of WHO guidelines are always in the market. I have just given one example of this part, how to discuss this thin layer chromatography. I think you can justify this by this also. The evaluation of the plant drug is done by studying its various properties. And we have studied all these in B form standard or M form standard. We have gone through different type of evaluation. But whenever we talk about the thin layer chromatography, ये slide दिख रही है मेरी?
Yes, sir. It's good. No, sir.
तो आपने देखा होगा यहाँ पे एक ट्रेन दिख रही है.
23rd, sir. Sir, it is 23rd.
ये ट्रेन दिख रही है?
23rd slide is showing.
ट्रेन दिख रही है?
No, sir. 24th slide is no.
अच्छा, अब दिख रही है?
Yes, sir.
ये ट्रेन दिख रही है सबको?
Yes, Professor.
थिन लेयर क्रोमेटोग्राफी मैंने लिखा हुआ है। मैं जस्ट आपको बताना चाहता हूँ कि रिसर्च करते समय या टीचिंग करते समय हम डिस्कस करते हैं कि पीएलसी इज अ वेरी कॉमन टेक्नोलॉजी फॉर इवेल्युएटिंग द हर्बल मेडिसिन और एनी मेडिसिन। हमने स्पॉट लगाए और चला दिया, बट हाउ टू नो what is the TLC? I personally feel that TLC is nothing but a very clearly comparable with this train. As you know, this train runs on this track, is it? Can you tell me, anybody, what is the speed of the rail? Average speed of the rail, anybody?
Sir, 70 to 90 kilometers.
Tell me. Rail never moves. Train moves. So rail is nothing but a stationary phase, and rail is a mobile phase, is it? इस स्टेशनरी फेज पर ये मोबाइल फेज मूव करता है, ट्रेन मूव करती है, और इसके अंदर पैसेंजर जो बैठते हैं वो कंपोनेंट, दे आर द कंपोनेंट। सो डिस्टेंस ट्रेवल बाय दिस मोबाइल फेज एंड डिस्टेंस ट्रेवल बाय द कंपोनेंट इज द आरएफ वैल्यू। एम आई राइट? बट स्टेशनरी फेज का यूज भी इंपॉर्टेंट है, क्योंकि यदि स्टेशनरी फेज का डिरेलमेंट हो जाए, यदि इसकी स्टेशनरी फेज सही ना हो, तो टीएलसी का आरएफ वैल्यू नहीं निकलेगी। दैट मीन मोबाइल फेज और कंपोनेंट मूव नहीं कर पाएंगे। So stationary phase, mobile phase and component can be compared by this train or rail or passengers also. And that can give you the RF value, because टिकट का जो दाम होता है वो उस पर डिपेंड करता है कि ट्रेन कहाँ तक जाएगी और कंपोनेंट कहाँ कहाँ उतर जाएगा। कंपोनेंट जहाँ उतरेगा उसकी आरएफ वैल्यू के हिसाब से ही, मोबाइल फेज के हिसाब से ही उसका टिकट लिया जाएगा। इफ द ट्रेन गोज टू कैलकाटा and the passenger stopped at Kanpur, that run by the RF value will be given by you, the Kanpur station to the train to Calcutta, and that's why the train is very good example for TLC. Am I getting the you getting my point? So evaluation of herbal medicine are different type. I'm just making this slide to give you some highlight. There are certain other examples are there. The fluorescence microscopy. Fluorescence microscopy can also be used for evaluation, where different type of starch grains, crystals of calcium oxide crystals, stone cells, and then vessels can give you a different illusion of the highlight or a different wavelength and different fluorescence identification, which gives you a very good example for the identification of certain microscopic compound in case of fluorescence light. Molecular marker. Nowadays, these are the different techniques we can use for the DNA marker, which gives you a polymorphism used for the DNA. And this is very important for the purpose of identifying the different metabolites, especially. Now, I'll earlier talk about the database. These are the very good example nowadays for the research people, that how to get a different type of database. PubChem, Drug Bank, Bioassay, then this therapeutical target database, Indian plant anti-cancer compound database, World Molecular Bioactive database, then herbal medicine database, and these database in the internet, you can get that detailed knowledge of the drug and you can use for your research. Presently, herbal medicines are applied widely, and researchers have growing interest in scientific proof and molecular analysis of herbal effects. And these database is widely useful for the researcher, those who are working on this part. Like in case of, there are more than 14,000 target of the drug bank database website are there. This database contains 6,796 drugs, including FDA approval, 134 FDA approved biotech compounds, 83 nutraceuticals, and 5,174 experimental drugs investigation drugs. Like similarly, in case of traditional Chinese medicine integrated database (TCMID), it records 47,000 prescriptions, 8,000 by 159 herbs, 25,000 more than 45 compounds, and disease are there, which can be useful for your further research and knowledge of particular drugs. In Indian IPA compound database, Indian plant anti-cancer compound is unique, providing the comprehensive information of phytochemicals of Indian origin with anti-cancer activity via 32 descriptive fields. So these databases are widely useful nowadays to start your research, on drug discovery, knowledge of a drug, whether the drug has been already published, whether the drug has been useful for different other disease or not, and this database can be widely useful for the uh, people, those who are working in the herbal medicine research, especially. Now, after going for different type of analysis, identification, extract, and isolation, people is still not thinking that whether they can use this drug for the different formulations or not, or whether your research or your formulation can be directly transferred to the industry. What are the major problem or the challenges of the industries are there? For that, I need to discuss few topics, especially in case of challenges in herbal industry, because research are going through, whether the people are working on pharmacology, whether the people are working in pharmaceutics, whether the people are working in pharmaceutics, in chemistry, and other quality assurance, they started their research from the pharmacognostic drugs from the herbal origin. But after completing with their research, there is no use to the society. There is no use for the industry. Here we are lacking. The research cannot be implemented in the drug formulation, and it cannot be come to the market because there is a little challenges in the herbal industry, because our herbal industry having lack of CGMP, no documentation validation product, lack of quality control procedures of the herb, where we researcher has to work much more and more. Although different pharmacopoeias are like Indian herbal pharmacopoeia, then new Indian pharmacopoeia has been comes out. So they are having a much more procedures are there, but still we need much more things or much more analysis to be required to validate for the herbal drugs. Documentation process and validations of herbals are very less. Batch to batch variation in herbal product, as I earlier told you. Toxicity profile is not explained. No scientifically approved efficacy and documentation are there in different pharmacopoeia. Properly designed quality assurance protocols are not there, and that's why the herbal industry is much more lacking in that case, and they are not producing the reproducibility of the quality of the drug, or they cannot provide assurance of the quality every time. So these are the important challenges always been for industries out there. Second, indiscriminate harvesting, poor post-harvesting treatments are there, because the people does not know about how to harvest the drug. They only go for how to cultivate. They don't know how to cultivate in detail. They don't know what is the right time of the collection. They don't know how to keep it in a storage, at what time, what temperature, where to keep it. Either in case of rainy season, they can collect it, and they cannot know how to make it dry. So the wrong handling of the medicinal plant, poor agriculture propagation method, and this may lead to a worst development of research, because high yielding varieties are not much more. So after getting a poor quality of drugs from the harvesting, from the field, from the farmers, you cannot go a better research. Inefficient processing technique lead to a low yield and poor quality product. Shortage of raw material, escalation of raw material prices, adulteration of raw material, lowering of standards of the product, and lack of R&D product and process development. These are the very important challenges the herbal industries are facing, and here the researcher has to work much more. where we can have to have a better knowledge of starting from the cultivation to collection to uh, quality control or quality analysis or phytochemical screening. I mean to say that researcher has to highlight on this part also. Now, after getting challenges, we should be think out that what we doing, what are the recent development the government and other industries are doing for. In 2022, IIT Delhi and Government of India with All India Institute of Ayurveda signed MOU to study the therapeutic benefit of herbal formulation and wellness. And that gives you an advantage that IIT Delhi having a good, much more in the institute, sophisticated instrument, and they started with this All India Institute of Ayurveda and getting a much more quality product. In May 2020, Ayush Ministry also collaborated with the Health Ministry, launched a clinical trial of ashwagandha, then Yestimathu, Gaduchi and Pipli and polyherbal formulation as known as Ayush 64 for the treatment of COVID-19. So these are the recent development where the government also look after the betterment for the herbal industry and herbal drug also, and the Ayush Ministry and the All India Institute of Ayurveda come forward to join hand with the government and doing better and better. In November 2020, Ayush Ministry approved the manufacturing of Asta 50. This is also one of the COVID-19 product which is taken care by the Dalmia Healthcare.
Sir,
Yes.
sir, sir, we have to rejoin the link for meeting is
खत्म
going
हो
to
गया
expire.
वैसे?
Yes, sir,
Okay.
we have to rejoin the link. All the
So
participants, please rejoin the link.
okay. I need my around 5 minutes more.
Okay,
ये
sir.
स्लाइड अभी आ रही है कि खत्म हो गया?
Sir, आ रही है अभी.
अच्छा अच्छा. So these are the recent development. WHO also started with India with a new training institute awareness of the Ayurveda, and then after announcement of Indian government inaugurated two Ayurveda institutes. In case of one is Jamnagar and another is Jaipur, with those who are working for the research of different herbal industries or different herbal products. And we are very hopeful for this ITRA and NIA. They are doing with a good job for better and better new herbal formulations in India. Should I join now? Dr. Sharita? Am I audible?
Yes, sir.
Yes, sir.
Okay, can you see my slide?
Yes, sir.
Yes, sir.
So I'm just concluding the part. So these are the recent development, the government of India and some of the leading organizations, including IITs and the ITRA, Jamnagar, and the National Institute of Ayurveda, has collaborated on different research in case of herbal medicines and Ayurveda, and they are come up with a different new technology and also new herbal formulation field of alternative medicine, where we can how the Siddha medicines or the Unani medicines can reach up to there, and they can go through a different technology, and they can discuss the different type of ailments and disease and their compounds also, and they isolated it and can use for the different clinical trial also. So nowadays we are in good progress, and researchers can use these type of knowledge for their own research, and they can go for different professions also. Very latestly, we are also coming with the part that herbal drug can be now converted into the nano formulations. And these nano formulations required whenever we have to have a very small quantity of drug for the betterment of the ailments. Because in modern medicines, now nanomedicines are being widely useful nowadays because they need a very little dose and it can be directly counteract to the disease. But in herbal medicines also, we are also gone for this nanomedicine. The global need for novel herbal drug formulations, like because they have a better bioavailability, improved stability, biocompatibility, dose reduction, where the herbal drugs are having a larger dose, एक पुड़िया दो पुड़िया छह पुड़िया 3 महीने तक खाना है. So novel drug, herbal drug delivery can have a dose reduction, minimize toxicity, and different type of other benefits are there. And for that, we need to have a herbal nano dosage form. We all know what are the materials are called because they are called smartness. I'm not going to detail. These nano herbal medicines can be nano capsules or nano carriers are there, and they can be useful same as per the modern medicine, where we can have entrapment of these drugs or maybe having a dispersed nanosphere. These are the certain nanoparticles are also in the market, and these nanoparticles, as all people are knowing, that these particles can be widely useful for with the herbal compound also, isolated herbal compound, where we can be very effective in case of drugs are being treated as a nano dose form. Different techniques for herbal nano formulation are being used nowadays. It may be ultrasonication, nanofabrication, precipitations, then spray drying, nano encapsulations are there, nano suspensions are there, and nano polymerizations are there. And these preparation methods nowadays are useful for herbal drug also. And these nano herbal formulations are in the market and very effectively they are useful by the patient. Few examples I have quoted it like Artemisia annua. Nanocapsules are in the market and they are very highly useful for the different type of disease. Artemisia annua is resistant uh, malaria, useful for the resistant malaria. Similarly, in case of curcumin, nanoparticles of curcumin have been prepared by the process of wet milling technique and useful for the different type of useful of. Similarly, in case of Brahmi by Himalaya, they are using the ketosine alginate nanoparticles, and these nanoparticles are very physically stable and useful for the different type of brain tumors. Berberin, nanoparticles berberin are successfully prepared by using single emulsion method or multiple emulsion method and can be useful in case of different disease. Genticin, isoflavin, nanoformulation of genticin is a market. Then paclitaxel, nanoparticles of paclitaxel useful in anti-cancer and widely useful by the modern physicians also. They are using this type of Genaxol in a market. Few herbal cosmetics are also in a market in a nano form. You can have this type of cream, lotion, gel, which are in very useful nowadays and can be useful for the persons also, those who are using this herbal drug in a nano form. Nano delivery system, polymeric and lipidic nanoparticles, these gels and curcuma face masks are there. Hair care nanoceuticals are also in the market, where the shampoos in a nano form cluster are in the market, which are anti-dandruff and also a good looking shampoos are there. Then some of the sunscreen lotions, which are being useful for the ultraviolet filters, and people are athletics, cricket people, football players, tennis players. are nowadays using this type of nano herbal sunscreen, which are in a market, and they are widely useful by the people. And it can be a benefit for the researcher, those who are publication, those who are doing this research in this herbal medicine in a nano form also. So this is my presentation. Thank you for all for listening carefully. If you have any question, you can ask me.
Thank you, sir, for such a nice and knowledgeable session about herbal drugs, its formulations and scope of phytopharmaceutical in the field of pharmaceutical industry, global herbal medicine market size increment, and its involvement in the growth, industrial growth in India, advances in analytical technology and biological science with many bioactive chemical entities from plant origin, various guidelines of regulatory agencies regarding herbal medicine such as the FDA, WHO, and in India, Ayush, IPC, various challenges in herbal medicine. Thank you, sir, for such a knowledgeable session. Participants, if you are having any questions, please you can ask. Anyone is having any doubt regarding herbal medicine? It is a nowadays it is a growing very fast. Okay, sir. One question is there.
Yeah.
Extraction method for sir. Extraction method for one plant. It that will be used. It will be same if it will be used for different purpose.
Extraction for a single plant.
Yes, sir. It will. Is it same for अगर सर सेम प्लांट को अगर हम दो कैटेगरी में यूज कर रहे हैं तो उसका क्या एक्सट्रैक्शन मेथड सेम हो सकता है या फिर इट विल हैव टू बी डिफरेंट
Extraction
एक्सट्रैक्शन
method
प्रोसीजर.
is always depending on the plant part, whether it is a leaf part, whether it is a root part, whether it is a fruit part or the flower part. First, so the
Yes, sir.
extraction process can be selected first on the part of the plant part, and secondly, what type of chemical compound is present in that phase, because the solvent should be selected first. If the solvent is if the compound having a polar compound, then you have to take the solvent, the polar solvent. If the compound is non-polar, then you have to go for a non-polar compound like chloroform or benzene or cyclohexane like this, or the precursor. If the polar compound like glycosides are there, like tannins are there, like polyphenols are there, then you have to select the alcohol or water or like that. So firstly, it is depending on which part of the plant we are using, and then whether the dry drug or the fresh drug is there, or then the solvent basis. Because if the heating is there, then the compound may be modified. It can be changed. Isomerization is possible. Then we have to go over some modern technique like carbon dioxide technology can be used. Some other emulsion techniques can be used. So it completely depending on what product we are using and what compound you thinking about that they have. So it is a process of review of literature. One day it cannot be decided.
Okay,
Or
sir.
there are
Sir,
different
one more.
techniques.
Yes, sir. Sir, one more question is there. अगर सर कोई
Hmm.
नया प्लांट है तो
Mhm.
उसके जैसे उसके अंदर कोई प्रॉपर्टी हमने फाइटोकेमिकल स्क्रीनिंग की और कुछ उसके अंदर कुछ ऐसे केमिकल्स हैं विच हैव सब मेडिसिनल प्रॉपर्टीज, तो क्या सर हम उसमें इन सिलिको मेथड यूज करके हम उसकी फार्माकोलॉजिकल एक्टिविटीज को सेफ्टी कंसर्न्स को आइडेंटिफाई कर सकते हैं या फिर उसके ऊपर रिलायबल कर सकते हैं इन
Definitely,
सिलिको
definitely.
मेथड
In
यूज
silico
करके?
method is nowadays very widely useful. कि आप पहले उसका ड्रग का कंपाउंड देख लें और उसको इन सिलिको करें, और इन सिलिको करने के बाद आपको थोड़ा हाइपोथेटिकली ये मालूम चल जाता है कि इसमें कौन से कंपाउंड किस तरह के हैं और उनकी बॉन्डिंग कैसी है, और उसके बाद आप उसको आगे बढ़ सकते हैं. Earlier it was
तो सर
not there, but
सेफ्टी वाइज
nowadays the in silico
सेफ्टी
is
वाइज
very
सर वो
widely
सही
used.
रहेगा?
Pardon?
सर सेफ्टी उसकी सेफ्टी और टॉक्सिसिटी जो है वो उस पे हम रिलाई कर सकते हैं उस पे
हाँ, बिल्कुल
इन
बिल्कुल
सिलिको
बिल्कुल.
मेथड?
इन सिलिको यदि हो जाएगा तो आपका एक पार्ट बहुत अच्छा हो जाएगा कि यू कैन जस्टिफाई ऑन द सेफ्टी पार्ट.
Okay. Okay, sir. Thank you, sir.
थैंक यू.
Now I would like to invite Dr. Shikha Srivastava ma'am to conclude for the session of this FDP.
Thank you, sir.
Thank
A very good afternoon to everyone. On behalf of Institute of Pharmacy, SRMU, I, Dr. Shikha, feel opportunity to propose a vote of thanks on this occasion. 1 week faculty development program organized between 5th to 11th July over the theme Advanced Research Methodology in Pharmaceutical Sciences has indeed been a very memorable event with such eminent icons of respective professions. I, on behalf of Institute of Pharmacy, would like to convey my deep regard and heartily thanks to Dr. Manju Singh ma'am from Pandit Ravi Shankar University, Dr. Pooja Chawla ma'am from ISF Moga, Saeed University of Health Science, Dr. Alok Mukherjee sir from United Institute of Pharmacy Prayagraj, Mr. Avinish Kumar sir from SRMU, Dr. Abhay Bharadwaj sir from KIT Ghaziabad, Dr. Ambar Vyas sir from Pandit Ravi Shankar Shukla University for attending this occasion and giving their valuable time for out of their busy schedule. Thank you for sharing and upgrading our knowledge with us and providing valuable insight over different aspects of research. I would also like to thank to fraternities from different institute and research scholars for their active participation. We are highly grateful to respected Professor Dr. Manju Pandey ma'am, Director Institute of Pharmacy for her wholehearted support and guidance. A special thanks to all the moderators of the session, Ms. Sanchalika, Mr. Neeraj, Mr. Sunil, Mr. Avnish, last but not least, Ms. Sarita Singh for solving all the technical glitch and handling all the feedback forms. And my special thanks goes to the administration members, teaching and non-teaching staff for their valuable support. At the end, I once again thank the distinguished assemblage in this FTP. Hope everyone would have benefited from this session a lot. Thank you all very much.
Thank you, Sarita madam. Thank
Thank,
you, Sikha madam.
thank you, sir.
And
Thank you,
thank you
sir.
to
A lot, sir.
Dr. Manju Pandey also. It was a nice
Yes, sir.
interaction
Yes, sir.
virtually.
Due to some issues, sir, ma'am is unable to join.
Okay.
Okay. Thank you, sir. So, thank you so much, sir.
Convey my thanks to Manju madam
Yes, sir.
because
Sure, sir.
she called me in, before starting this part. But on 8th, I have a PCI inspection. That's why I cannot continue on this 8th or 9th. So I make it this day for a better presentation. So if you have any questions, anybody from any researcher or any teacher, if you have any query or inquiry, you can take my mail from Dr. Sarita Singh. So you can interact with me anytime. I am available for that any query. Thank you.
Thank you so much, sir.
Thank you, everyone. Thank you for making this a successful event. Thank you, everyone. Thank you so much.
Okay. Thank you, ma'am.